Welcome to the second webinar organized by the Chromosome 6 project, a project that was started upon request of parents, is sponsored by parents, and conducted together with parents in order to improve quality of life of individuals with a chromosome 6 disorder and their families. The first webinar was organized to announce the launch of the International Chromosome 6 Foundation. In the present webinar, we will briefly summarize the aims of the webinar series and of the Chromosome 6 project. The main part of the webinar consists of two topics. Firstly, we will explain to you how you can read and understand the results of a microarray report. Secondly, Eliana will present to you what we have learned about the clinical effects of terminal 6p deletions. Terminal means that the deletions are at the end of the chromosome. At the end of the webinar, we will explain to you why we need your help and input. This really is a project by and for parents. As mentioned, you can ask general questions in the chat, and we will try to keep an eye on them. Directly after the webinar, we will move to another private Teams channel where we will answer questions of parents who have received a separate link via Pauline. The aim of the Chromosome 6 project, Chromosome 6 webinars are threefold. We want to inform you, the parents and other people interested in the care for individuals with a chromosome 6 disorder. We want to create awareness for this group of often ultra-rare diseases for which still, notwithstanding all new developments and technologies in genetics, hardly any information is available on what to expect clinically and how surveillance can best be organized. And thirdly, we want to invite you to actively take part in this project. We need your support. We need your help to collect more information to make our interactive information system more robust, informative and reliable. We need your feedback on how things can be made better in the Chromosome 6 project for you, parents. We need to know how we can improve your participation and how we best present the automatically generated information that our system will provide to you. Lastly, but vital, we need your help for financial support in order to further develop our system and secure its future existence. In this slide, we have summarized the main members of the Chromosome 6 project team. If you want to know more about us, please watch webinar 1. That is still available at the website of the Chromosome 6 project. The project team is located at the University Medical Center Groningen in the Netherlands. This is an accredited center of expertise for rare chromosome disorders. The team consists of clinical geneticists, psychologists, bioinformaticians, and PhD students. Other medical specialists can be consulted whenever needed. Now I will briefly summarize the aim of the project. Again, for more general information about the Chromosome 6 project, please watch seminar 1. The aim of the project is to have more information available for families on the effects of chromosome 6 aberrations on health and development. And we hope that by having more information on natural history and comorbidities of these extremely rare disorders, that we can empower families, improve surveillance of the individuals, and improve quality of life. This really is a study initiated by and performed with parents. We recruit patients via social media. We collect information directly from parents, and this is done with our multilingual extensive questionnaire. We use microarray reports that have been uploaded by parents. We have worked hard on a very sophisticated automatic analysis of the collected data. It's almost ready to be presented to you. And our final goal is to develop an interactive web page where parents can obtain a phenotype prediction of each chromosome aberration. Now today I will talk about the microarray report. In order to better support us, 3 Dutch parents founded the International Chromosome 6 Foundation initiated by Pauline, who you see on the upper picture together with Roland. Since this is an international foundation, parents from other countries are needed to take part as well, and some already have volunteered to do so. If you're interested as well, you can email the foundation using the email address on this slide, info@chromosome6foundation.org. Now, let us start with a very practical but also very theoretical part of the webinar, how to read a microarray report, sometimes also called a SNP array, array CGH, or just simply an array. In order to explain a microarray report, I first need to explain chromosomes and the different ways chromosomes can be studied. Visually, by looking through a microscope, or quantitatively, by measuring the amount of DNA, the material that forms the chromosomes. I will explain what can be detected by microarray and what not, and finally how to read the report. This slide summarizes how our genetic information is stored in the cells of our body. So what you see here is a human being, and each human being consists of millions of cells, and each cell, I'm trying to use my pointer, but it doesn't work, unfortunately. So you have to believe me, this, you see there in blue, that is one cell, a figure of one cell, and in the middle of the cell you see is something that's called nucleus. And in that nucleus, our genetic information is stored, and it is stored in the chromosomes. As I will show you in the next slide, there are 46 chromosomes in each human cell. A chromosome consists of a very long string of DNA, but you can unwind, and then you see the DNA is made out of what we call base pairs, and that are what's also mentioned on the side as nucleotides. A simpler word is base pairs, and these bases are 4 different bases, and the order of these bases make up for your genetic code. By looking through a microscope, the chromosomes can be seen and counted. In this way, aberrations in the number of chromosomes and large deletions and duplications can be detected. To detect smaller aberrations, the so-called submicroscopic deletions and duplications, another technique is needed, the microarray technique, as I will explain in a minute. To detect changes in the genes themselves, so changes in the genetic code, then the genetic code has to be read. This is called sequencing. The order of the DNA building blocks or base pairs is determined by a technique called sequencing. So you can't see this order of the base pairs by microarray. You need a different technique, sequencing. Normally, everyone has 46 chromosomes in all the cells of the body. And these chromosomes, they can be ordered from the largest chromosomes to the smallest chromosomes, and then you can see that these 46 are made up of 23 pairs. So you have all chromosomes double. One is coming from father, the other from the mother. There's a slight difference between men and women. Women usually have 2 X chromosomes, and men have 1 X and 1 smaller chromosome, the Y chromosome, and the Y chromosome contains the information that is needed to become a man. Now, I need to explain what chromosome bands are and how they are numbered. As you can see, this is a schematic drawing of chromosome 6. When you look through a microscope and you color the chromosomes, you see all these dark and light bands in the chromosomes. And these have been numbered. And it depends a little bit on how beautiful your chromosomes are, how well all these little bands, how well you can see them. So that's why this numbering of the bands is a bit strange. First of all, each chromosome shows a constriction somewhere in the chromosome. You can see it by each chromosome. That's called a centromere. It divides the chromosome in a small part and in a large part. The small part is called the P arm, short arm. P is French for petit. And a large part that's called the Q arm, the long arm. And here in these arms you see the numbering from the centromere outwards. So this is band P12. And if you see a small line in between, you can say, oh, this is 12.1, 12.2, 12.3. It's very strange numbering. Then you have band P21, P22, etc. until 6P26. And in Q, it's the same. So that's how we number the chromosome bands. If we look through a microscope to the chromosomes, this is what we see. A bunch of bars with dark and light colored bands. Luckily for us, the chromosomes can be ordered automatically. A camera on top of the microscope makes images, and the computer program sorts the chromosomes 2 by 2. Then next, a technician studies the chromosomes in detail, and sometimes For instance, a small deletion can be found. You can see here the top of this chromosome is missing. Now, what is a microarray? Actually, it is a glass slide. It is a glass slide with rows, also called arrays, of small spots with unique pieces of DNA with a known chromosomal location. So each of these tiny dots that you see here on the slide contains DNA, a very tiny stretch of DNA, which is actually a very tiny part of the chromosome. It's a unique code in there that it belongs to that tiny part of the chromosome. So if you know from each dot with DNA where it belongs on the chromosome, you can use that to measure the amount of chromosome that is present in a patient. And I will explain that in the next slide. So on such an array, there are a few thousand ultra small pieces of chromosome 6. This glass slide can be mounted in a so-called hybridization chamber. That's what you saw on the, go to the previous slide if that works. Here, what you see here, this is the hybridization chamber, and you can put DNA on top of the glass slide in this chamber. Go to the next again. Okay. So let me briefly explain to you in a simplified way how the microarray technique works. Chromosomes of the patient and of a control, sorry, I need to click, you can see it, okay. Chromosomes of the patient and a control are cut in tiny pieces of DNA. These pieces of DNA are labeled with a fluorescent color, either in green or in red, and then the DNA of the patient and the control is added in equal amounts to the glass slide with the array in this hybridization chamber that I showed you on the previous slide. Now, DNA is a very sticky substance and it is able to recognize itself. So, the DNA of patient and the DNA of the control want to stick on the DNA in the spots. And for each spot, we know where it belongs to the chromosome. So, we can measure the amount of red and green DNA in each spot. That's what we do. We measure it and then we calculate how much DNA there is of the patient compared to the control. In this graph, you see the result for chromosome 18. The dots in between the dashed lines, so these dots, they represent that says that, okay, here in these spots we see equal amounts of patient and control DNA. That's a normal situation. At the lower end of the chromosome, less patient DNA is present. So there seems to be a loss of the lower part of chromosome 18. And of course, this is for an example of chromosome 18. We can do the same for chromosome 6 and for all other chromosomes. So usually when we do a microarray in a patient, we look at all the chromosomes. Are all these spots in between the lines, and are there equal amounts of patient and control DNA? And I'm going to show you examples of that. Chromosome 6. Okay. Too much or too little DNA of a specific part of a chromosome is called a copy number variant. Too few or too many copies of that chromosome part are present. When there is a loss of genetic material, then that is a deletion. You can see an example here for chromosome 22. This is chromosome 22. These are all the spots that belong on chromosome 22, and you see here there's a drop, a loss. And when I enlarge that part, you can see here, this is the normal amount of DNA, and here is a loss of patient DNA. So this patient has a small deletion at the end of chromosome 22. You can also see a gain of genetic material. That's seen over here for chromosome 7. Again, this is chromosome 7. Here you see a small gain. When we enlarge it, it's very tiny, or a very tiny gain at the end of chromosome 7. It can be that there's more than one copy number variance in one person. And this actually is the case here because this patient had a deletion of chromosome 22 in combination with a duplication of chromosome 7. So you can have multiple copy number variants in one person. It's important to know that not each copy number variant will have an effect on the health and development of a person. If a deletion or duplication causes clinical problems or causes disease, we call this a pathogenic copy number variant or pathogenic CNV. But we also know that there are benign CNVs. That are copy number variants, so losses and gains of genetic material, of chromosomal material that have no clinical effects. Sometimes we see that a copy number variant has what we call an incomplete penetrance. Sometimes it causes an effect, but other times it does not cause an effect. And in lay terms, we call that a risk factor. Mostly when you have such a risk factor, there is an enlarged chance of learning problems, but you do not need to have learning problems. And also these variants can run through families. They're often inherited from a healthy father or mother. Then last, we have copy number variants, from which we do not know what the effect is. We call that variants of unknown significance. These are the most problematic ones because if we find such a copy number variant, we do not know what to tell the parents. So what can be detected by microarray? Not all genetic aberrations can be detected. What can be detected are aberrant number of chromosomes or small and large deletions and duplications, losses and gains of DNA stretches. What can not be detected? Well, a deletion of a complete gene can be detected by microarray. Tiny change in the genes themselves, so a change in the genetic code, cannot be seen. As I explained to you before, if you really want to see where the base pairs are, you need another technique that's called sequencing. For instance, whole exome sequencing, and most of you also know that term. But with the microarray, you cannot see mutations in genes. Changes in the chromosomes that are balanced, so the normal amount of chromosomal material is still present, can also not be detected by array. You see here an example of a translocation where part of a chromosome has been translated to another chromosome and vice versa. The total amount of pink and purple chromosome is still present, so no copy number aberration is present. This is called a balanced translocation, and while there are no gains and no losses, the net amount of DNA is still normal. You cannot detect it by microarray. You need another technique. Actually, you need to look through the microscope to see this. Sometimes a chromosome can form a ring. This is an example of a ring chromosome 6. The outer part of the chromosome at both ends, so at the P arm end and at the Q arm end, is then missing. This loss can be seen in the microarray. So you see that there is a loss at both ends of the chromosomes, but you cannot see that it is due to a ring formation. So if you see that in an array, always the laboratory asks, oh, let us do a microscopic study. Let us look at the chromosome 6 itself because we think this might be caused by a ring chromosome. So that's when the microscopic evaluation still is important. Now, you all have all the background information, so let's move on to the microarray report. Here you see an example of the conclusion of a report. I will explain in 2 slides what this long text string means. So, 46XX. The child has a normal chromosome, the normal number of 46 chromosomes with 2 X chromosomes, as is commonly seen in girls. R means that the study has been done using an array technique. So microarray, SNP array, array CGH, array, all these terms can be used, but it is an array technique. Then in between square brackets, you can see HG19 or something, another code is put there. And that means that this is the number and the version of the human code as it is known at the moment of investigation. Your child's DNA is compared to what is called the reference DNA, what is known at that time of the DNA code. 6q27 means that the chromosome that has changed is number 6, and the chromosome band is number 27 on the long arm Q of the chromosome. So here again, chromosome number 6, and the deletion is located here. And I know it's a deletion because of this, and that I will explain in the next slide. Okay, here it is. So this bunch of numbers says, followed by ×1, says that, okay, these are the base pairs. So the DNA consists of a long string of base pairs. All these base pairs are numbered from the top of the chromosome downwards. And in this example, the base pairs 166,609,388 until base pair 170,919,470 are only present once, ×1 in the string, instead of twice, the normal situation. So there is a loss of genetic material, a loss of chromosomal material, a deletion of 4,310,082 base pairs. In short, 4.3 million base pairs, 4.3 MB. Okay, then the last thing in the text string is the letters DN, and that stands for de novo or new, which means that it is not present in parents. It will only be in your array report if the array has not only been done in your child, but also in the parents. So in summary, doctors call this a deletion 6q27, located from position 166.6 to 170.9 megabases on chromosome 6, with a size of 4.3 megabases. Sometimes a result can be very complicated, as shown on this slide. You see in a text string that an array was performed. You see R. You see also in between the square brackets which reference DNA was used, and that there are 3 CNVs. The first one on the short arm of chromosome 6, 6p25.3 to p25.1. It's in the green square on the right side of the slide. There's a second, and that is again, it's ×3. Then there is a loss, so deletion in 6q27, so the red square in the slide on the right side. And in addition to that, there's also an aberration of chromosome 9 in band Q3.1 ×1, so a loss on chromosome 9. Okay, let me explain to you what this means. The duplication has a size of 4.2 megabases. And we know that these kind of duplications results in low birth weight and motor delay. The deletion is smaller, but because the deletion is genetic material missing, it has a more severe effect than the duplication on 6p. These are listed here are the clinical features that are commonly seen in these kind of deletions, and in one of the following webinars we'll talk more in detail about this, but you can see there's a lot of features that can be present when you have the deletion of that part of chromosome 6, like a small head size, dysplatic ears, hypertelorism, which means wide-spaced eyes, vision problems, dental abnormalities, feeding problems, etc., etc. So a long list of features that can be present. They not necessarily all have to be present, but these are the features that we commonly see in patients with a terminal deletion of chromosome 6q. Now, the last copy number variant was the deletion 9q. It's very small, only 0.2 MB. It's benign. It has no clinical consequences. So this is a known benign CNV. Okay. Now, this is an example of a variant of unknown significance. As I explained before, we do not always know what the effect of a CNV is. This is an example of such a variant of unknown significance. It is a duplication in the short arm of chromosome 6, a very tiny one, only 0.3 megabases. The report gives also a bit of explanation about this. It says, well, it's a duplication from 70.7 to 18.0 MB. It's small, but there are a few genes in this duplication. The function of these genes is not known yet, so we also do not know whether extra copies of these genes have a harmful effect. It has not been described in literature to cause clinical problems. It is also not present in the database of normal variants. We have a database, it's an international database, where all normal variants are stored, so that we know if we find something, that we can see, is it known as a benign variant or not. It's not in that database of benign variants, so we do not know yet whether this very tiny duplication can cause clinical problems or explains the clinical features of the child in which these chromosome studies were performed. Okay. This is my last example that I show you because in a minute Eliana will tell you more about terminal 6p deletions. Here you see a deletion 6p that is almost terminal. It's not quite on the top, but it's almost there. We call that subterminal. The indication for chromosome studies in the child was cleft lip palate, cleft feet, micropenis, and wide-spaced eyes. The error result is depicted here. You see the chromosome bands again. These are again the base pairs that are deleted because it's ×1, so it's a loss of chromosome material. And it says a deletion 6p25.1 to p22.3. So this in this part over here, it's 9.2 megabases, pretty large. And this causes a 6p deletion syndrome. That's also in the report. They don't only show in the report what they found, but also what it means. And here the reporters say it causes the 6p deletion syndrome with developmental delay, hearing problems, eye problems, then the Walker malformation, which is a brain malformation, kidney problems, heart defects, cleft lip palate, and short neck. So the cleft lip palate of this child is explained by her chromosome aberration. Now, I hope that with this information, you are able to decipher the micro result of your own child. If you still have questions, just discuss this with your doctor and be aware that also for many medical specialists, array reports can be complicated and the clinical geneticist is needed to understand it in full detail. Okay, time for our next important topic, the effect of terminal 6p deletions. For that, I give the floor to Eliana. Eliana, go ahead.
Thank you, Connie, and thank you everyone for joining our second webinar. As Professor Connie already said, I will talk about the health-related consequences of 6p25 deletions. And I'd like to start by going very quickly through what are 6p25 deletions, even though Professor Connie already explained quite a lot of it. And these are deletions that involve the last part of the short arm of chromosome 6. The last three bands of chromosome 6 are called 6p25.3, p25.2, and p25.1, and they are collectively known as the 6p25 bands. Deletions that involve any of these three bands or all of them are called 6p25 deletions. Now, if we zoom in on this part of the chromosome, we will see that the 6p25 region, like many other regions, has many genes, and these are only a few of them, just to give an example. The very last gene on 6p is called the DUSP22 gene. Deletions that also involve this gene, so deletions that extend all the way at the end of the chromosome, are called terminal deletions, while deletions that are in the 6p25 region but do not include the last gene, so they do not extend all the way at the end, are called subterminal deletions, or you can remember it the way Professor Connie said, almost terminal deletions. As with other regions of chromosome 6, we wanted to know what are the health-related effects of 6p25 deletion. So what does it mean for someone's health if they have a 6p25 deletion? So in other words, we wanted to study the phenotypes of these aberrations. And by phenotypes, I mean all the medical issues, developmental problems, behavioral, and other characteristics that an individual will develop. So why is it important to study the phenotypes of these aberrations? First of all, in order to provide you, the parents, with information that can prepare you for any issues that your child might develop in the future. Also, that you can be able to explain why your child has certain features that are different than other children. The second reason is that knowing the health-related effects that can be expected in a child can help healthcare providers give more suitable advice regarding the type of care, management, follow-up that a child might need. So this information allows affected children to receive better care by their doctors. Our study on the phenotypes of 6p25 deletions was conducted at the end of 2022, and it got published in a scientific paper this year, which you can find online. Last week, we also published a summary of this paper on our website for parents. If you go on our website and then on results, this will be the first hit that you will get. And in this summary, we also explain, we also include the link to the original paper if you'd like to read it or discuss it with your healthcare provider. And in the summary, we also explain how to read the different tables on the paper, and the paper is open access, which means anyone can access it online for free. I will now also give a summary of our results. All right, so first, what did we do to describe the phenotypes of 6p25 deletions? At the time of our study, 13 parents of children with a 6p25 deletion had fully completed the chromosome 6 questionnaire. And we also found 46 individuals with a deletion in that region that had been described in the medical literature over the years. So we also collected information from the papers on these individuals as well. We used the medical and genetic information from these 59 individuals to make a description of the medical issues and developmental abilities of children with a 6p25 deletion, also called a clinical description. Now this figure shows the deletions of all individuals included in our study, and it looks a bit complicated, but I will walk you through it. On the top, you can see chromosome 6, and the part that is in the red box is what is zoomed in the rest of the figure. You can see also the 6p25 bands, which continue in the beginning, and after the 6p25, you can see the rest of the bands in the P arm. Yes. Every horizontal bar in this figure represents the deletion of one child. The red ones are deletions of individuals that are participating in the Chromosome 6 project, and the black ones are deletions of literature reports, of patients described in literature. For example, if we look at a red one, that will be a Chromosome 6 project participant that misses that part of Chromosome 6, while a black bar is an individual described in literature that also misses that specific part of 6p25. Now, you can see from this figure that all these individuals had deletions that included at least part of the 6p25 region. Some had very small deletions that only extended to band 6p to 5.3, like the one on top. And others had much larger deletions that included the whole 6p to 5 and other bands further down the chromosome. The vertical lines, the vertical blue lines, represent important genes in this region. We call these genes important because, according to studies, if these genes are deleted, patients will likely develop some medical or developmental issues, so it is essential to know if any of these genes are included in someone's deletion. One very important gene in this region is this gene called FOXY1, which you can see there, and it is almost at the end of the chromosome. And you can see that almost all individuals in our cohort included the deletions of all individuals included that gene, so the gene FOXY1, all but one, so except for the one deletion at the very top that had a very small deletion. What you can also see in this figure is that all of our study participants had deletions that were quite different in size. Some had tiny deletions and others had very large ones. And this made it difficult to generalize the features of these individuals and use them to describe any 6p25 deletion. So in order to be able to study their phenotypes, we split these 59 individuals into 6 groups. And this splitting was based on on the deletion size and on the important genes that were included in their deletions. And you can see on the right the 6 groups that were made. Subgroup A only included one patient, and that is because there was the only patient that did not miss the important gene FOXY1. And then subgroup B had deletions that were terminal and extended up to 3.15 MB or million base pairs, as Professor Cohn already explained. Subgroup C had 5 individuals that were with deletions that were slightly larger, so up to 4.02. Subgroup D had 13 individuals with deletions, terminal deletions up to 7.11 MB, and subgroup E had the largest terminal deletions, so larger than 7.11 MB in size. The 19 individuals in our cohort that had a subterminal deletion, so they did not extend to the end of the chromosome, were grouped separately. What we did is we looked at the phenotypes of each subgroup and the whole group and made a description of the features that were seen more commonly. Yes. So what did we find? First, some general characteristics of our study participants. From the total of 59, 36 were females, 20 were male, and for 3 individuals described in literature, we did not know their biological sex. Their ages ranged from 1 month old to 53 years, with a median of 6 years and 6 months. And what this means is that half of our group was younger than 6 years and 6 months, and half of it was older. So this shows that most of our study participants were children, even though we did have some adults. Now, if we look at their phenotypes, we noticed that they had many common features, independently from the subgroup at which individuals were in. So individuals in group B, which had relatively small deletions, had many common features with individuals in subgroup E with the largest deletions. This shows that the FOXY1 gene, which was present in deletions of almost all individuals, plays a very important role in the kind of features that a child with a 6p25 deletion will develop. I will now give an overview of these features. But before I do that, I want to ask you to keep in mind that every child is unique. Not all children with a 6p25 deletion will have all the features that I will mention, and they might also have other health-related problems or medical issues that are not described here. This is just a summary of features that we saw most often. So this is a list of features that were commonly seen, and by commonly, I mean they were present in at least half of the total group. And I have categorized them into 4 main groups. The first group is problems with the eyes, vision, and hearing that were seen in the majority of individuals. And around 75% of children in our cohort had problems in the development of their eye, specifically a defect named Axenfeld-Rieger anomaly. And this anomaly is a spectrum of developmental problems affecting the foremost region of the eye, also called the anterior segment. And what I mean by spectrum is that it includes a range of problems. Some of these problems you can see easily from the outside, such as in this image here, where you can see that the black color part of the eye is not developed properly, or it is split in multiple parts, or it is displaced. But certain variations of Axenfeld-Rieger might require a special eye exam to identify it. If you click once more on here, you can see that in this case, you can see a ring around the iris, the color part of the eye, and this shows that that part is thickened. So there is also a variation of Axenfeld-Rieger, but any variation of Axenfeld-Rieger anomaly is quite characteristic of 6p25 deletions or other genetic aberrations involving the FOXY1 gene. Besides these developmental eye problems, children in our cohort often also had squint eyes and sometimes had involuntary eye movements, what we call nystagmus. Most of them were farsighted and they needed glasses for that, and the majority also had mild to moderate hearing loss. Now, on to the second group, which is heart and brain defects. These were seen also in the majority of the group. Brain defects were seen in around 60 to 70% and heart defects in about half. And a very characteristic brain defect in 6p25 deletions is an abnormality called Dandy-Walker malformation, which affects the cerebellum, which is the smaller part of the brain located at the very back. And this part, thank you, Connie, this part is important for movement, coordination, and balance. On the left side of this image, you can see how a healthy brain looks like without defects. And the right side shows a Dandy-Walker malformation. And you can see that in this case, the space between the skull and the cerebellum, which is normally quite narrow, here is filled with fluid. And in this case, part of the cerebellum is also not developed properly. Now, Dandy-Walker malformation has also different variations with different severities. And the effect that this malformation will have on brain function or on movement, on balance, really depends on the specific abnormality. For heart defects, what we saw most common were defects that were affected mostly the walls between the right and left side of the heart, what we call the septa. So these are called septal defects. In the small box, once back. Yes, in the small box up there you can see a healthy heart with an intact septum, and on the left side of the image you can see that there is a hole between the left and the right side, and this would allow blood to flow from one side of the heart to another. And this type of heart defects also vary in severity. Some defects are very small and they can cause no symptoms. They might go unnoticed even, and they might close on their own. And some of them are large enough to require surgery. Some other types of heart defects that we saw were defects of the heart valves. And these are the small muscles that help push blood around in the heart or from the heart to the rest of the body. We saw that the majority of our study participants also had low muscle tone, and around 80% of them also had developmental delay, which was mostly mild, but I will give more details on development later. Children in the group also had special characteristics of the face. These are two of the children that we included in our study, and you can see that what they have in common is the fact that their eyes are spaced a bit further apart than typically. And you can see in the lower left picture that the ear is positioned a bit lower than normally. And you can see also in both of them that the part of the cheekbones is a bit more flat. Next to these features, children also showed abnormalities in the formation and development or number of teeth. So here I have included a list of symptoms that were not seen as commonly, but I have added them because these symptoms are clinically important to know, which means that might affect health or require treatment, and for this reason are important to know. So what we saw is a variety of bone and joint problems that in most individuals, but mostly they included hyperextensible joints, scoliosis, which is a sideways curvature of the spine, an abnormal position of the foot, and other bone deformities, mostly of the longer bones, which are the bones affecting the arms or the legs. About a third of patients also had an increased eye pressure, what we call glaucoma, and this was almost always a consequence of the Axenfeld Rieger anomaly, which is known to cause an increased eye pressure. In some children, the middle and inner ear was not formed properly, and all these children also had hearing loss, but we could not find a pattern between the presence of these problems and how severe the hearing loss would be. So we cannot say that if a child has an abnormal middle inner ear, he or she will have more severe hearing loss. Some parents also reported that their children got frequently sick with infections. However, we should say that this is something that we see often in children with a chromosome aberration, or more often than typically developing children, and that is independent from the chromosome or the type of aberration that they have. Now, lastly, 6 individuals reported to have experienced seizures in their life, and 3 of them had diagnosed epilepsy. Now, once more, all these features were seen relatively often in individuals in all subgroups, so we can say this phenotype description is characteristic for a 6p25 deletion, independent from group deletion size. Now, in individuals with terminal deletions that exceeded a certain size, so they were slightly larger, and namely larger than 4.02 MBs, so those that extended beyond the green line in this figure, subgroups D and E, these individuals had some additional features next to the common features that we just discussed. And these features included feeding difficulties that persisted after the newborn period, a cleft lip or cleft lip and palate, which is a split in the lip and or palate, kidney problems, mostly kidney cysts or an underdeveloped kidney. And what we also saw is that individuals with larger deletions, if they had a heart defect, then they had more than one heart defect or more severe heart defects, which we call complex heart defects. Now, on to development. 83% of the total group had developmental delay, and the delay varied in severity. Developmental delay was quantified using IQ or DQ. I think everyone knows what IQ stands for, but DQ stands for developmental quotient, and that is a score on development that compares development of children to typically developed children. But what we saw is that 7 individuals had a typical development and 4 had borderline delay. And when developmental delay was present, it was mostly mild. However, for 13 individuals, we knew that they had developmental delay, and these were mostly literature cases, but we did not know how severe the delay was. For 11 of them, they were too young, so namely younger than 2 years of age, and we could not say whether they would go on to develop typically or not. Developmental delay, when present, was mostly global, which means it affected motor, speech, and cognitive abilities. And we also could not find a relationship or a pattern between the size of the deletion and the severity of developmental delay. So we cannot say that the larger the deletion, the more severe the delay would be. However, what we did see is that typical development and borderline delay was seen only in individuals with smaller deletions. So if we go one slide back, we saw that those 7 individuals with typical development and 4 with borderline delay were only in subgroups A, B, and C. Individuals in subgroups D and E always had developed some degree of developmental delay as far as we knew. We can go back. Yes. So in the article and in the summary on the website, you can find a table with the ages at which children reach certain milestones, such as walking without help or speaking, also in comparison to children without a chromosome aberration. I think we can go to the next one. Yes. So this is a table that is included in the paper and gives a number of recommendations for medical checkups that should be considered when a child is diagnosed with a 6p25 deletion. And those are described in the column which is called upon diagnosis, and also recommendations for medical investigations that can be considered during follow-up visits to the doctor under the follow-up column. I will now go quickly through this summary, but you can find this table both in the summary on the website and in the paper. So we recommend that any child diagnosed with a terminal or subterminal 6p deletion should have a thorough neurologic investigation, including a brain MRI, to check for brain defects. An EEG should be considered if seizures are suspected. And all children should also have a thorough eye exam done by their ophthalmologist or an eye doctor, including measurement of eye pressure. The hearing should also be tested, and an ultrasound of the heart should be done at least once to check for heart defects. We also give some recommendation on what your doctor should pay special attention to during regular visits. For example, tooth abnormalities, growth, or posture. And of course, development should also be assessed. For terminal deletions that are larger than 4.02 MBs, we have some extra recommendations since these individuals had some additional features. And these recommendations are that they should undergo also an ultrasound of the kidneys, and special attention should be paid to the lip and palate development. Now, to conclude, individuals with a 6p25 deletion that include the gene FOXY1 have a common phenotype, which means that they have many common health-related features. And this phenotype seems to not be dependent on the size of the deletion. Individuals with a terminal deletion larger than 4.02 MB have some additional medical issues. Of course, every child is unique, and the clinical consequences of their 6p25 deletion will vary. Lastly, but very importantly, with this study, we showed once more that collecting information directly from parents and families can help us better understand the health effects of rare genetic disorders and offer better care to affected individuals. I would like to extend a special thank you to the parents and patients that participated in the study and made the study possible, and of course to the rest of the team. You can find a summary of the paper and the link to the original paper on our website. And if you have any questions, you can, of course, add them in the chat or email them at chromosome6@umcg.nl. Thank you.
Thank you, Eliane. I think this information nicely shows what our project can do for families and doctors. In our subsequent webinars, we will discuss different parts of chromosome 6, so please stay tuned. But we also want to discuss more general aspects, like we did today with the micro report. So the webinars will remain informative for everyone. So not only for those with a 6p deletion, 6q deletion, or somewhere else, other part of the chromosome. In each webinar, we also want to give some general information. And please contact us if you have ideas for us on what to present. So if you have any questions, you say, oh, please explain this in more detail to us, then we'll make a webinar for you. For our next webinar, we already posted on Facebook this question. We asked you, what tip or recommendation would you like to give to other parents or families that has been life-changing for you? Something that you changed that really, for instance, your child has very severe sleeping problems and you had this very good tip that finally your child slept in a better way, or feeding problems or other things that really helped you and say, okay, this is something that I would like to share with other people. We made an online form where you can anonymously give these tips. We have said, okay, do a max of 3 tips per parent, and the deadline is December 20th, so that, well, during our next holidays, we will be able to make an overview of all these tips, and we will give them in the third webinar, and that third webinar is scheduled for February 3rd, 2024. So, and of course, not all tips will be applicable to every child and every family, but I think there certainly will be something in there for you. Now, we're almost at the end of this webinar. As mentioned at the beginning, the Chromosome 6 project is a project by and for parents. We are a group of dedicated researchers and parents. We really do this together. We collect our data from families worldwide, which is really great. And in this way, we have been able to create the largest chromosome 6 database ever. We have already shown how helpful this is. We have very good recent publications that show that, okay, we can give recommendations for better care for these children because simply by the fact that we know more. We know better what how the development is, what the behavioral issues are, and what medical comorbidities arise. And hopefully soon our dream will be realized and we will be able to give you interactively on our website detailed patient-specific information directly to you, the parents. Allow me to summarize how you all can get involved in the Chromosome 6 project. And I know I also did it in webinar 1, but I think this is very, very important. You can participate in the project if your child has a Chromosome 6 deletion or duplication. Just go to our website and you can see how you can do that, or drop us an email at chromosome6@umcg.nl. You can advocate the project, for instance, by becoming a member of the International Chromosome 6 Foundation Advisory Board and be an ambassador for the foundation in your country. If you want this, please send an email to info@chromosome6foundation.org. And lastly, you can support the project by crowdfunding, for which information can be found on the project's website. In our recent letter to the Facebook group, we explained that 55,000 euros is needed to secure the continuation of the project. Since our first webinar, we raised over 3,000 euros. This is a great start, so let's continue this good work. Simply share this QR code within your social networks. Ask people to scan it and donate whatever they like. All little bits will help because together we can make this happen. Now there is a short video that Pauline sent in to us because she now made it possible that via the Facebook group you can start funding actions. And so she made a video to explain that. And I'm asking now, Eliana, can we show that video?
Yes, we can.
So I stop sharing my screen?
Yes.
Thank you, Eliana. I'm sure this short movie, which is very instructive but very short, it will also be available on the Foundation's website, so that, if it went too fast for you, just go to the website and look at it again and see what you can do. It's very easy to create a fundraising activity on Facebook now. You can share it in your social network, and who knows, with the holidays coming in, people are enthusiastic to help us out. Good. So with this last slide, I want to say goodbye to you on behalf of the whole team. For the parents who have been invited, I'm happy to meet you using the other Teams link within 5 minutes, so the private meeting with the families. I will leave this slide, the next slide here, for a couple of minutes, so that if you want to make a picture of it or scan the QR code, please go ahead. And now with that, I'll ask Eliana to stop the recording and to go to the other meeting to allow the parents in there.